playing with the embryos like Zivi.
Very good.
Very good.
What's the time in Florida right now?
It is 1 o'clock, 1 p.m., 13 hours.
1 p.m.
Okay, let's start.
Let's chat, אוקיי, אז בואו, אנחנו נתחיל כמה דקות.
ליאור, נתחיל?
כן, נתחיל כמה דקות. א', אני רוצה להגיד תודה רבה, רבה, רבה ליניב, ותודה לכם שאתם ככה מצטרפים. אני עוד שנייה אעבור להאזנה, ואני חושב בכלל שאנחנו ככה צריכים לעשות את זה יותר, ככל שאנחנו נלמד אחד מהשני, וככל שאנחנו נבין טוב יותר את הענף שאנחנו, את המקצוע שאנחנו עוסקים בו, ככה התוצאות לא ירחקו לבוא, נכון? זה לא שעה נוחה לאף אחד, ובכל זאת זה מאוד מאוד חשוב. אנחנו באירוע ענפי לא קטן בנושא של החמאה, בעצם לפני הוציאו את החמאה מפיקוח, יש לזה משמעויות לא קטנות מבחינתנו. אנחנו מניחים שגם בשבועות הקרובים שר האוצר יחליט לגבי הסיפור של המכסים על פטור ממכס, וגם מהבחינה הזאת יש לנו משמעויות לא קטנות, אבל בסך הכל אנחנו עובדים תחת הסכם, ואנחנו מצפים שלמרות ההתבטאויות שככה כל שוק החקלאות ככה נמצא באיזה סוג של שייק, אנחנו מצפים שיעמדו בהתחייבויות כלפינו. אז זהו, שיהיה לכם ערב מענה ממש, ויניב תודה על הזום. תודה על הזום, וחברים, אני רק ממש מבקש לעבור כולם למיוט, אין לנו פה את עדי שעושה את זה, אז אנחנו לא הרבה אנשים, אז שכל אחד יהיה אחראי על עצמו.
Okay, then good evening everybody. Happy to see a lot of people, and again I want to thank Jos'e Santos that is being here with us today. I will again say please be everybody on mute. We'll have the beginning, we have the presentation, and then after the presentation we'll have time for discussion. Okay, then, שומעים? Can you hear me?
Yes.
Yes.
Yes, okay. Okay, then Jos'e Santos is a professor in the Department of Animal Science at the University of Florida in the USA, where he conducted research and extension in dairy cattle nutrition, health, and reproduction. Jos'e received his DVM degree from Sao Paulo State University in Brazil in 1992, then complete the MSc and PhD degrees at the University of Arizona, USA. Then he did clinical residency in the dairy production medicine in 2000 in the School of Veterinary Medicine in the University of California, Davis, and complete sabbatical at the University of Sydney in Australia. He spent 8 years as a faculty member with clinical and research responsibility in the School of Veterinary Medicine at the University of California, Davis, before he moved to the University of Florida in 2008. Jos'e has been a major professor of 17 PhD students and 24 master students that was a guide of. He has hosted more than 10 postdoctorates and sabbatical visitors and another more than 100 visiting students. He has published more than 250 manuscripts in a peer-reviewed scientific literature, 14 book chapters, and has given more than 400 presentations in technical and scientific meeting. Jos'e seeks to develop technology that enhance efficiency of dairy production, particular improvement in prepartum health and reproduction. The research is highly integrative, combining components of basic cellular biology, all animal physiology, and applied intervention that are adopted by dairy producers worldwide. Jos'e Santos was here in Israel in our conference in Jerusalem in 2010. We remember, and he was there, and they were very happy to have him here today with us. And Jos'e, I want to give you the floor. You can share the screen presentation.
Okay, well, thank you, Yaniv, for the kind introduction. So I hope you can see my screen. I'm going to bring this up, and I think once I do that, I will no longer see you, but hopefully give me a second so I can work through this thing here.
Yes.
Okay, I'm going to duplicate this so hopefully it works. And let me see if I have if I remove this here, hide floating. Okay, well, I guess good evening, everybody. It's a pleasure to be here, although it's remote. But I just upload a copy of this presentation in the chat box, so everybody can have a copy of that. Hopefully you can access and open it. And my goal is really to discuss what has happened in the last probably 10 or 15 years in the US dairy industry relative to reproduction, which is the focus of this discussion here. So I'll tell you a little bit about a lot of things that you already know very well, but hopefully I'll characterize some things that I think have been quite interesting and quite important that hopefully justifies where we are today. Okay, so a little bit I'll talk about the driving forces for what we have seen the last few years, which is an improvement in dairy cow reproduction. And then as opposed to picking on particular details, okay, I want to spend a little bit of time on the big picture, on the things that have probably a major impact. And I have chosen to focus on these three areas here, okay? There is a fourth area, which is this, what I call the hidden element or the age factor. And that is the human element, which is something that for me as you know, somebody who works with cows, it's a difficult one for me to characterize, but I think it's very important. I see this on farms. The farms that really do well in multiple aspects are those that have an open communication between upper management and the staff who actually execute the tasks. So this human element here, which I find very difficult to characterize in research, seems to be a pivotal player in the success of many, many farms. So I'm going to tell a story more like this, you know, in a sort of a simple system, but the reality is that the story is quite more complicated. And it took time for us to learn how to do things right. So things don't happen, you know, new technology that are available. It takes time for us to learn how to use properly and to know exactly what to expect from that. And I'll spend a little bit of time on that. Well, as you probably know, we live, you know, at least in the US, but probably worldwide, a very volatile environment for pricing. If there is one thing that producers have no control, is milk prices. They can plan, but they really don't control that. Okay, so producers are what we call price takers, at least in the US and in the world market. And you can see, you know, over the years, what has happened here. So this is milk price in the US when we had the 2008 crisis, the year that I moved to Florida. Milk prices averaged 25 cents per liter. Okay, obviously well below production costs. And then we had times of just outstanding milk prices. This is when people go broke, farms close, some people committed suicide. This is when people become rich or buy the next farm. Yeah, so we live this sort of environment here. So if you look today, our milk prices here in the Southeast, which is a different milk market in the US, it's approximately 40 cents per liter, depending upon milk composition. But this would reflect the federal milk marketing order, about 37 cents per liter, pretty close to what the price is today. So it's a very volatile market, and that creates its own problems. Obviously, like in the Israeli dairy industry, the US dairy industry has a successful story of changing the cow that we work with. This is a plot of a lactation curve of a dairy cow in the US in the 1940s to 1950s. This was the observed milk production. So very mediocre cow. You know, you could see that's a small cow, a very different prototype of animal. So if you look at the body weight of that cow at the time, this would be the potential for dry matter intake if that cow behaved like a cow of today that has this pattern of increased intake, reaching a plateau, and then eventually decreasing. Okay, obviously, I really don't know what the intake was because people didn't measure that and report in scientific manuscripts in detail. But if you look at the cow today, she is more like this one here. This shape, if we average lactation curves of multiple cows, okay, it would look more like this. Obviously, individual cows don't necessarily have this shape of lactation curve, but the average of a group of cows would be more like that. So, yeah, through genetic selection, we have changed, you know, this cow in terms of body size, the ability of the mammary gland to secrete products, and that has changed the lactation curve. But we have not been able to change directly feed intake. And that creates a problem for reproduction because, you know, cows prioritize production early lactation. And because we select for that, we can measure the phenotype very readily, but we don't have, you know, phenotypes for dry matter intake that we could have selected cows for. Therefore, we create this period of catabolism and tissue mobilization that poses a challenge for reproduction. So if we pick the extreme of the population in terms of phenotype, I think this cow exemplifies that. So to my knowledge, she's still the world record today. She produced almost 36,000 kilograms of milk in a lactation, but perhaps more impressive is the amount of milk solids that she produced. Okay, so approximately 2.5 tons of fat protein combined. And if you do a little bit of math, this cow here averaged almost 100 kilograms of milk with pretty solid milk fat content and true protein content. So this would be an animal that produces the equivalent of 3 cows today, yeah, on a daily basis in terms of milk solids. So almost 7 kilograms of fat plus true protein per day continuously over the course of a year. So obviously she's the exception, but to be honest, when we go to large herds, we do find cows, not necessarily like this one, but we find many cows that produce at least half of what this cow produced. Okay, so it's not uncommon to find cows that average 18 to 20,000 kilograms of milk in a lactation, and I suspect that we're going to have more and more of those cows as time goes by. Okay, so has that been an issue for reproduction? I think it's a challenge, but let's just look at the data from the national herd today. The US has about 9.2 million dairy cows today. It has dropped somewhat, about 100,000 cows in the last year. And the Department of Agriculture collects data on approximately 2 million of those cows that go through our national record system for phenotypic evaluation for milk production monthly, and then it collects reproduction data. So here we have pregnancy per artificial insemination in all AIs in the lactation. And I broke this down into two breeds, the Holstein cow and the Jersey cow. Jerseys or Jersey Holstein crosses represent about 12% of those 9 million dairy cows in the country today. Holsteins represent almost 8 million dairy cows in the country today. So you can see that starting somewhere in the early 2010, we've seen a progression or an improvement in pregnancy per insemination in the Holstein breed. Not really a change in the Jersey, but an improvement in the Holstein. If we look at the first AI only, where we typically see the best pregnancy per insemination, we observe about a 6 percentage point increase in pregnancy per AI. So this has shown to me that in spite of the continuous increase in production, somehow the industry has been able to capture an improvement in pregnancy per insemination. And obviously, the combined improvement in pregnancy per insemination with reproductive management programs that optimize insemination rate, we have been able to decrease calving interval in almost 2 days per year. So we are sitting now at about 396 days of calving interval. And this value is not really biased by culling decisions or by, eliminating cows from the herd. So this has really been a successful story, at least in the last 15 years or so, of a continuous decline in calving interval, meaning that our days open have been decreasing in the last few years steadily, small numbers, but steadily. So we are in a sort of a upward improvement progression at this point. So we have passed the bottom of the worst of reproduction, and now we are in the upward of reproduction, at least for the Holstein breed. Probably the Jersey is a little bit of a different story because producers put less emphasis on reproduction in Jerseys because they perceive that breed to be a little bit better than the Holstein relative to the ability to become pregnant and maintain pregnancy. Well, so let's, a few years ago, I collect data from the Eastern United States, and I broke down herds based on the metrics that we use to evaluate reproduction, which is what we call a 21-day cycle pregnancy rate. And I'll show you exactly what this is so you understand how we measure that. Okay, so just to give you an idea, the US herd today is sitting at about 18.5 to almost 19% pregnancy rate, okay, which is a combined insemination rate with pregnancy per AI. So I divide the US or the Eastern US herd into very mediocre herds and outstanding herds, okay? These herds that have above 28% 21-day cycle pregnancy rate, these are herds that in 3 periods of 21-day cycles after the voluntary waiting period would get cows pregnant. So in about 60 days after they start breeding, cows would become pregnant. So the bar graphs here represent the rolling herd average. So the average milk production per cow in a lactation, okay? And you can see that these herds with above 28% pregnancy rate, okay, they have, when I collect this data, they have an average annual production over 11,000 kilograms, so about 11,500 kilograms. And the average pregnancy rate in those herds was about 31%. And there are more than 2,000 herds here. Okay, so this is telling me that there is a large group of producers that learn how to produce a lot of milk per cow and have excellent reproductive performance. So we need to learn from these people. Okay, so what is this pregnancy rate that I'm telling you? So this is how we measure reproductive indices. So the first one is estrus detection rate. Okay, it's probably better represented by insemination rate. For every 21-day cycle, so an interval between 2 estruses, it's expected that open cows will return to estrus and they should be inseminated. So insemination rate is nothing more than the number of cows detected in estrus, or better said, the number of cows inseminated divided by the number of eligible cows to be inseminated. These are cows that have passed the voluntary waiting period of the farm. They are not pregnant, and the producer wants to breed her. The second index is pregnancy per AI, also called conception rate. This is what I would call the inheritability of the cow to become pregnant once she is inseminated. So it's the number of pregnant cows divided by the number of inseminated cows. So this tells a portion of the story. This tells another portion of the story. But the third one, which is what we typically measure, is what I call 21-day cycle pregnancy rate. And this is a combination of the numerator of this index with the denominator of the other index. So this is nothing more than the number of pregnant cows divided by the number of eligible cows that could become pregnant, whether we bred her or not, okay? So obviously we measured this at 21-day intervals just because of the simplicity, but the reality is that this rate is daily. So we breed cows every day. But in terms of farm calculations, or what producers are familiar with, or what the national herd uses, is a 21-day interval. So let's just look at this from a herd perspective as an example. So imagine that the voluntary waiting period of a farm is 51 days. So starting at 51 days in milk, cows become eligible to be inseminated and become pregnant. So in this first 21 days interval of breeding, I have 100 eligible cows, or I have 100% of the herd eligible to become pregnant. Imagine that in those first 21 days, 60 cows were inseminated. So my insemination rate, or estrus detection rate, is 60% in this case. Imagine that 24 cows out of the 60 inseminated became pregnant. So my pregnancy per AI, or conception rate, is 40%. But the pregnancy rate, which is the 24 pregnancies divided by the potential 100 pregnancies, is 24%, okay? And if I do this for every 21-day cycles, and I truncate it here at 155 days in milk, I have the overall estrus detection rate, the overall pregnancy per AI, and the overall pregnancy rate in this example herd here. At the end of so many cycles, I would have a total of 335 possible inseminations, okay? Because an open cow is expected to be inseminated every 21 days if she read the book. But the reality is that this farm, in this example, only performed 176 inseminations. Therefore, the insemination rate is 46%. This farm here obtained 58 pregnancies. So it's 58 pregnancies out of 176 inseminations. So that would average 33%. However, this farm obtained 58 pregnancies out of a total of potential 335 pregnancies opportunities. So our pregnancy rate truly is 17.3. So this is the number that we typically look. Again, the US herd phenotypically is sitting about almost 19%, but I gave you examples of herds that are sitting at above 30%. So why do we worry about this? Well, obviously reproduction has an economic benefit to the farm. That's why we focus on that. It can affect milk production because it affects the distribution of the herd along the lactation curve. Okay, so if I improve reproduction in general, I reduce average days in milk of the herd, so I move my herd to a phase of the lactation in which I capture more milk per cow per day. It can now the parity distribution of the herd. So if we think of first virgin heifers and first lactation animals, we can move them faster to a more productive lactation, such as the second lactation, in which we capture an additional 15 to 20% production. But it can also increase the number of replacement heifers. So this creates opportunities for more genetic selection pressure, so greater intensity of genetic selection. So we can pick and choose what heifers we keep. because now we have more of them available, okay? In some markets allows us to sell heifers and cows for milk production. It doesn't really apply today to the US industry because it costs us today to raise a heifer about $1,600 until she gets to 23 months to calve. But if I want to sell that heifer in the market, I don't get much more than $1,300. So today we want to have a small heifer inventory just because of the current economics. But this creates a lot of flexibility. Having better reproduction allows me to make more wise culling decisions. Now I don't get rid of cows because I'm forced to, and I don't retain cows because I don't want to have an empty herd. Now I can have more educated decisions when I make culling decisions. So that creates opportunities for income and for better management. Okay, so this is a plot of a lactation curve of one of my former clients' herds in California. So there are about 6,000 cows here, and this is based on daily milk production. This is the projected dry matter intake curve. Obviously, we never measured this one here. We measured the milk, but based on the milk and the average size of the cow, I basically projected how much those cows had to eat to support this level of milk yield at different stages of the lactation, okay? And here are some of the inputs I used for the price of milk, for the price of feed. And then I calculate income divided by feed cost, which is this line here. So you can see that earlier in lactation, my income over feed cost is greater than later in lactation. And that is related to this persistence of lactation and how much the cow eats, okay? The two components, if we don't play too much with the price of the diet. So having a herd that has fewer days in milk, it's better relative to income over feed cost than a herd that has more days in milk, okay? Because now I have more money left to pay all the other expenses, such as the fixed cost, the heifer inventory, labor, et cetera, okay? So what is the impact of that on milk yield? So when we project a reduction in days open, just for ease of calculation, from approximately 15.4 months of calving interval, okay, to about 13 months of calving interval. If I cut this calving interval by about 60 days, which is a huge change, I can capture another 1.5 kilograms of milk per day of calving interval, just because of the persistence of lactation. If we were to have a flat lactation curve, then we would not capture a lot more milk, yeah? So this reduction in calving interval would result in an estimated 500 extra kilograms of milk per cow per year in this herd that averages a little over 12,000 kilograms of milk in a lactation. So it does have an economic impact above and beyond having replacement heifers, so on and so forth. It affects the milk production per cow. Well, one of the challenges of cows is that, you know, in life there are priorities. Although we like to think that reproduction is a priority, it is for the species, but it's actually a bad thing for the survival of the individual. And there's lots of wildlife data exemplifying that, even cattle data in Australia from Queensland showing that if a cow is pregnant during droughts, she dies faster than a non-pregnant cow. And she dies very fast during a drought when there is lack of nutrients when she is in advanced pregnancy. So it's no surprise that the body creates conserved mechanisms that when cows eat enough, she stores nutrients. Okay, she stores carbohydrates as glycogen in the muscle, in the liver, or she stores fatty acids in triacylglycerol in fat depots. But then she moves that and mobilizes that when she needs, like in early lactation, for example. But here the priority is to make sure that she survives, is to maintain what I would call essential processes, okay? So maintenance requirements of the animal okay, ion transport, walking activity, body temperature, circulation, etc. And then she have what I call reducible processes. She eats more, she produces more milk. She eats less, she produces less milk. And then lastly is what we focus on, which is reproduction. Okay, it's an expendable process. There has to be signals coming from the gut, from the viscera, from the gastrointestinal tract that interplays with signals from the hypothalamus to control reproductive cycles. And I think we know very well that those signals are in place in the cow as well as in other species. Let me just show you a little bit of epidemiological data to show the importance of this preservation of body tissues and reproduction. So these are data from almost 6,000 cows that we synchronized to receive a first AI at about 70 days postpartum. And we collect blood samples to determine whether they were cyclic by sequential blood progesterone analysis. And then we inseminate them and they either became pregnant or not. So there's lots of things in this statistical model. I'm just going to show you two things. One is body condition score change from calving to 65 days in milk, okay, as a proxy for more catabolism or less catabolism. And we can see the cows that lost more body condition score in a 1 to 5 scale, the traditional body score 1, 2, 3, 4, 5 with quarter points. Okay, fewer of them were cyclic compared to cows that didn't lose very much body condition score or lost and regained in the first 65 days. Okay, now if we look at milk production, we see no relationship whatsoever. Mathematically, cows that produce more milk in the first 90 days in milk, and this is quartiles of milk production within parity. So here I have the lowest primiparous and the lowest multiparous, the production of those cows in the first 90 days in milk. And here I have the best primiparous and the best multiparous, the largest quartile in the first 90 days in milk. So a difference here of 18 kilograms of milk per day. And you can see the ability to be cyclic, if anything, mathematically goes up. Okay. Now, if we look at pregnancy, it follows the same pattern as cyclicity. We have more anovular cows here, we have fewer pregnancies. We have more cyclic cows here, we get more pregnancies. And we see no relationship phenotypically between milk yield and the ability to become pregnant to a first insemination. So this tells me that we need to be sure that we don't have too many cows that undergo extensive catabolism in early lactation. So if energy balance, or what I would call nutrient balance, is important for reproductive success, we should focus on nutrient intake. So we should do things that makes cows to consume the amount of nutrients that they need, because energy balance explains most of the variance, or energy intake explains most of the variance in energy balance. So here I have data from 160 cows that we have daily milk yield, daily milk composition, daily body weight, and daily body condition score. So we calculate energy balance in the first 60 days in milk. And you can see that as intake of energy increase in early lactation, think of dry matter intake, energy balance improves. But if I look at energy balance as a function of the amount of energy secreted in milk, okay, think about milk production or energy-corrected milk production. Okay, the relationship is negative, but the R square is minimum. Yeah, so it's very small. So this variance that we see in energy balance is very poorly explained by the amount of milk secreted by the cow, but it is extensively explained by how much the cow eats. Okay. Well, if we think mechanistically how this cow is getting ready to be bred again, okay, so we have to go back to basic physiology or neurophysiology. And here I have a drawing of the base of the brain, in which I have the hypothalamus and the hypophysis here, just sitting below the brain. Okay. And it turns out that those tissues require sugar. as the primary energy source. So they require carbohydrates, either for the peripheral cells around the neurons or for the neurons themselves that get fed, the intermediates of the TCA cycle after sugar undergoes glycolysis or after glucose undergoes glycolysis in the peripheral cells. But it turns out that when cows calved, blood sugar concentration drops. So typical pattern of early postpartum cow. So if you look at this value here, it's sitting right at 3.6 millimolar, and then you have this hyperglycemic spike right at calving, and then this cow's drop in blood sugar because now the mammary gland has priority in sucking up glucose for milk synthesis. At the same time, this cow is adapting herself to provide nutrients for milk synthesis, so she undergoes adipose tissue catabolism, and I'm pretty sure she will undergo some degree of muscle catabolism and mineral or skeleton catabolism in early lactation. So this scenario here has to send signals to the brain to reduce the ability to resume astrocyclicity depending upon what state this cow is. So let's look at an example of a proof of concept type of experiment and use the ewe as a model for the cow. So this is a cool experiment in which ewes had their fourth ventricle of the brain catheterized so people could infuse things in the brain of the animals. And what this group here did, they infused either saline as the control treatment, or they infused 2-deoxyglucose. So here's a molecule of glucose, okay, the 6-carbon energy source, carbohydrate source. And here's a molecule of 2-deoxyglucose that has a substitution right in this position here, okay? So the hydroxyl group of glucose in the 2-carbon is replaced by a hydrogen. And this replacement here stops the glycolytic pathway. Therefore, the cells in the brain cannot extract ATP through glycolysis. Therefore, energy availability to the cell is reduced. And you can see here when they measure LH pulsatility in those ewes, infusing saline really didn't change the pulsatile pattern of LH secretion, but infusing 2-deoxyglucose abolish LH pulsatility. So this is telling me that A, if there is less nutrient available to be oxidized by the cell, the brain has some mechanism that tells the animal to secrete less GnRH, LH, and that probably impairs resumption of ovulation. So when I think of this as a mechanism and as a behavior of cow, this is typically how I see it. If an animal is well fed, she has sufficient amount of oxidizable metabolic fuels. So think of carbon sources, short-chain fatty acids, long-chain fatty acids, maybe some glucose itself, amino acids, okay, that gets processed by the body, and it provides carbon source for the hypothalamus. This supports the release of GnRH, which travels to the pituitary that supports the release of gonadotropin. And these gonadotropins are going to set the stage for ovarian or gonadal function. So this cow now has follicles developing that are steroidogenic, and the brain is capable of responding to these steroids such that this cow now express her typical behavior when she is cyclic, and hopefully eventually she ovulates, okay? And we hope that this will happen before the end of the voluntary waiting period. But now we have individuals that undergo a period of extensive catabolism because they probably don't eat very much, or they have competing factors that utilize nutrients, and we'll see one of them. Now, the hypothalamus, this cow doesn't see enough of these oxidizable fuels, and this blunts the signals to support gonadal function with gonadotropins. So now this cow has no steroid-dependent reproductive behavior, so she doesn't show estrus. She has no surge of GnRH, LH, therefore she has no ovulations. She undergoes a period of long anovulatory period postpartum. She becomes what we call today an anovular cow, okay? So I'm going to tell you a little bit of things that I think affect the ability of this cow to resume proper reproduction. Okay. So here's a study that we finished recently, in which we looked at cows in 2 herds over 3 consecutive years, and we model the risk of developing disease, production performance, and reproductive performance according to days in the prepartum group. So farms in the US, we have taught producers to design groups, design diets, and take care of cows when they are in the last 3 or 4 weeks of gestation. So if those things work, cows should benefit from that, but they need to spend time there. Okay, and we typically tell producers that they need to spend 3 weeks there, 3 or 4 weeks in the prepartum group. So we asked the question, is that true? Is that the sweet spot for days in the prepartum group? And what we found was this quadratic relationship between days in the prepartum group and the probability of cows to become pregnant in the first 300 days postpartum. And you can see that for both multiparous cows, cows that will start their second or greater lactation, as well as for nulliparous cows, cows that will start their first lactation, the highest probability of pregnancy was observed when they spend 3 to 4 weeks in the prepartum group. At the same time, the highest milk production in the subsequent lactation was also observed as when they spend 3 to 4 weeks in the prepartum group. And to a large extent, this was driven by reduced morbidity. Well, sort of a make or break stage in the life of a cow is this moment here. And we tell students, vet students, that you need to be patient, you need to be clean, and you need to use a lot of lubrication. This will define whether we start a lactation poorly to some extent. Okay, sort of a game between saving the calf or tearing the cow and causing trauma in the vagina that leads to metritis, which is a very important disease because it explains most of the morbidity in early lactation in US Holstein herds. So this cow that starts her lactation, she goes from a period in which intake is quite marginal. She's eating here 10, 11 kilograms of dry matter. And as soon as she lactates, her dry matter intake has to increase sharply. And this is a very cool study with multi-catheterized cows by Chris Reynolds in England. So you can see the level of production of those Holstein Friesian cows. So they peak here at about 45 kilograms of milk. The beauty of the study is that they had catheters placed in the portal vein, mesenteric arteries, and the hepatic vein. So they could look at net flux of nutrients. And I'm going to show you one figure here so we can appreciate the changes that take place between 3 weeks before calving and 4 weeks postpartum. So a short period of about 2 months or 1.5 months, we see a doubling in blood flow through the liver. Most of this blood flow is coming from the portal vein. And we see a 5 to 6 time increase in extraction of nutrients by the liver to support lactation. So this cow in a very short period of time has changed substantially. And if you think about cows that produce today 60 or 70 kilograms of milk, this number here would be much smaller than that, or much more negative, I should say, larger number, more negative, to support the mammary gland. So it's no surprise that if we make mistakes with those cows, they are going to suffer more than a less productive animal. So let me show you a very interesting experiment, but it's with beef cattle, but it uses the same concept. These animals had catheters placed in the portal vein, in the hepatic vein, in the mesenteric blood vessels. So they could look at net flux by using a marker, a dye that's used to measure blood flow, and then by measuring the concentration of nutrients, using venous arterial difference, you can calculate the extraction or the release of a nutrient. But the cool thing about this experiment is that they imposed treatments. So they had steers that were kept as controls. Okay, so they received saline in the trachea. And they had animals that were induced to develop an inflammatory response. So they infused bacteria, Mannheimia hemolytica, the old Pastorella hemolytica, in the trachea to induce a systemic inflammatory response. They were interested in respiratory disease in steers, but they were looking at nutrient balance in those animals. Okay, so I'm going to focus on the comparison between no inflammation and inflammation. But I'm going to make a parallel between this steer here that has respiratory disease with this postpartum cow that has metritis. Okay, and hopefully you will be able to see my video here. So this is lochia of a cow that has normal postpartum. So it is mucoid, is reddish because of blood that's present in the uterus, but this discharge is mucoid. So this is pretty typical. It doesn't smell extremely bad, but this is a cow that has metritis, okay, what we call metritis. Okay, it's a systemic, or it's a localized inflammation that often is expressed as systemic inflammation. Now she has a different flora in the uterus that causes this watery type of discharge, often with presence of tissue debris, you know, perhaps some pus or not, but maybe some chunks of blood or endometrium lining that's being sloughed off, okay? And this cow has really foul smell because of the anaerobes that grow in the uterus, and this makes her be very sick. Some of them even die, and I'm sure many of you had the pleasant experience to get this discharge sprayed on your body, and your wife won't allow you to go home. So anyways, so this is a bad disease that has the hallmarks of systemic inflammation in about half of the cows. There's increased acute phase response. Half of the cows will have fever. They show depression, they eat less, so on and so forth. Okay, so let's look at the data from the steers. So I'm going to show you one figure. This is the flux of amino acids across the liver, okay? So the line here represents a neutral net flux, meaning that the amount of amino acids that come to the liver, either from the portal circulation or from the arterial supply through the hepatic artery, is exactly the same as the flux of that particular amino acid that leaves the liver through the hepatic vein. Okay, so you would be right here at the zero line. If the number falls below the zero line, it means that the liver extracts more than it secretes. If the number is above the line, it tells me that the liver now is synthesizing that amino acid, so it extracts less than it secretes into the hepatic vein. So the first two bars represent the essential amino acids. So think of methionine, lysine, histidine, arginine, okay? So amino acids that typically are not synthesized by the tissue because the tissue lacks essential enzymes for those pathways. You can see, to no surprise, that the numbers are negative. So the liver has a negative net flux of essential amino acids. This is expected, that's the typical biology. But you can see it's more negative in the animals that were induced to have inflammation. If we look now at the non-essential amino acids, those that can be synthesized by cells through transamination reactions, now we see a positive net flux, but only in the animals that were in the control group. There's still a negative net flux in the challenged animals. So if we superpose this is what we see. Okay, for this 400 kilogram steer, we see a difference per day of about 2.6 moles of amino acids. And just for simple calculation, I use the average molecular weight of the 20 proteinogenic amino acids. That translates into approximately 380 grams of circulating amino acids in the bloodstream. Think of metabolizable protein that escapes the liver and is now in the circulation. So if you use the efficiency of memory extraction of amino acids of 67%, that's the protein equivalent to synthesize protein in 8 kilograms of milk. So you can see that this animal is changing how it uses nutrients if undergoes an inflammatory challenge. Unfortunately, too many of our cows become sick in early lactation. Okay, so this is when we diagnose metritis in cows. So it's right here between days 5 and 7, the peak of prevalence of metritis in dairy cows. If we look at non-uterine disease, so disease that affects the gastrointestinal tract, like DAs, diarrhea, bloat, affects the mammary gland, mastitis, affects the legs of the cow or the hoof of the cow, okay, lameness. Most of it takes place in the first 3 weeks. So if I superpose the first 3 weeks with the first 21 days, I have this ugly picture that 30 to 35% of the cows are affected by disease in the first 3 weeks of lactation. And almost 80% of the first disease diagnosis in most dairy farms take place early on. So this has a legacy effect that affects survival, production, but also reproduction in the cow. So let me show you, the impact of disease on different stages of reproduction in the cow. So here's a study in which we synchronize a little over 900 cows on a commercial farm, and then we flush the uteri between days 5.5 and 6 after insemination. These are single ovulating cows. So we recover almost 420 embryo oocytes, and we looked at fertilization, embryo quality cell number. And then in the second study, we synchronize cows, inseminate them at about 60 days in milk, but now we flush the uteri between days 15.5 and 16. And then we look at the embryo shape, development size, and a few other things that I'm not going to get into a lot of detail. Okay, so this is the 420 embryo oocytes. So we had diagnosed whether cows had disease or did not have disease. So these are cows that we didn't diagnose clinical disease, and these are cows that we diagnosed clinical disease. So they represent about 40% of those cows. And you can see that if we look at cleavage as a proxy for fertilization and then embryo quality, at any stage we look, cows that had disease before that first insemination, they had a lesser ability to establish and have a pregnancy. If we look now at day 16, the conceptus is smaller, so it has some retarded development, and that conceptus produces less interferon tau, which is important to prime the mother cow's uterus to not secrete prostaglandin. Therefore, she maintains the corpus luteum, and hopefully the embryo survives. So you can see that very early stages of development, pregnancy is already reduced if a cow had a disease event. Now, in a subsequent study, we transfer embryos to cows or inseminate them on a commercial farm. So there's about 4,000 cows here. And we had previously diagnosed them whether they had or did not have disease. And diseases are divided here in cows that were healthy to the best of we could characterize before first AI or first embryo transfer. Cows had only non-uterine disease, so disease that affects tissues other than the reproductive tract. They had only uterine disease, so retained placenta metritis in this case, or they had both. So there's about 4,000 cows for first insemination, and half of them receive an IVF embryo, half receive artificial insemination. And you can see that it doesn't matter what type of disease, or I should say, it doesn't matter what tissue is affected, pregnancy after first AI or first embryo transfer is reduced. And it gets a lot worse if a cow has both disease. A lot of it because pregnancy loss is increased by twice the risk. So the losses of pregnancy between the initial diagnosis of pregnancy all the way to calving, more than double in cows that had metritis combined with mastitis, for example. Okay, so that's a major problem. So given that we have a little bit of an idea what are the challenges, let me tell you a little bit what producers have implemented, sort of a broad, big picture to improve reproduction in cows. So hopefully I can get this quickly in the next 10 minutes so we have time for good discussion. So a major emphasis has been put into the environment of the cow. If you go to newer farms, they are built to state of the art in terms of cow comfort, cow flow, employee access, and comfort at multiple levels. Comfort in terms of where the cow is, comfort in terms of environment, temperature, okay? So methods for cow cooling, which is pretty important. People have implemented proactive prevention and therapeutic strategies, such as dietary changes that are implemented. So now diets are much better. Therapeutic strategies are usually evaluated through science-based experimentation to define how we treat cows. Okay? We have management practices during the transition period to minimize disease. Not always successful, but that's what's implemented. We have reproductive programs to optimize pregnancy per AI and pregnancy rate. And we have implemented heavily genetic selection programs to improve health and fertility. So I'll go quickly through some of that. So farms are just better today than they were 20 years ago. They are cleaner, they are nicer, cows are more comfortable, people are better trained, cow comfort, cooling, whether it is a compost barn or whether it's a traditional freestall or whether it's a tunnel ventilated barn, they are just better today than they were 10, 15 years ago. Okay, so here's somewhat of a timeline in the life of a cow through the transition period. Cows get dried off when they are about 230 days of gestation, and we hope that we accomplish a few tasks in this period here. Then when they are about 255 days of gestation, we move them to the close-up group. And we hope that the farm is able to properly group cows. And then here, there's a major focus on feeding diets that minimize metabolic disorders. Milk fever, displaced abomasum primarily. I think we've learned a lot and we know how to control those two diseases, but we still have a major challenge when it comes to calving here, and we don't know how to control very well metritis. That's something that we need to learn more. What is the solution to reduce the risk of metritis in cows? We treat too many cows because too many of them get sick. Then in early lactation, we feed diets that hopefully don't limit intake, such that cows don't lose too much body tissue. And we measure this by assessing subclinical ketosis as a proxy for tissue catabolism. Okay, so if we have too many cows with subclinical ketosis, we know that our transition program is not very effective. So we have too many cows losing too much body fat. Okay, but hopefully once she graduates from this period here, we feed diets to maximize production and recovery of body condition. So this is sort of the life on the average farm in the US. And all of this is done with proper cow comfort and heat abatement. And we've learned a lot from your industry, from work done in Israel, as well as in the US, that we need to cool cows throughout the lactation. From the day that she goes to the dry period to the day that she's dried off again. So she's always provided with proper cooling. We need to have a breeding program, and our breeding program relies on two technologies: detection of estrus and timed AI. Okay, so good producers use both and take advantage of both. But there are some goals here, okay, some milestones that need to be accomplished. The first one is that producers want all their cows bred within 3 weeks of the voluntary waiting period. So that's the first thing. The second thing, they want somewhat short inter-AI intervals. So in the good farms, the interval between breedings usually sit at about 30 days. Okay, and you can only do that if you combine aggressive detection of estrus with synchronization programs and routine visits to diagnose non-pregnant cows. Most farms start breeding in about 60 to 80 days in milk, and the goal is to get about 55 to 60% of the herd pregnant by 120 days in milk. That's where it is today. It may change as we learn more the economics of reproduction. So let me show you two examples. Here is a scatter plot of a herd that is very poor in terms of managing first AI. So here I have days in milk at first AI, and here is the day postpartum of the cow. Imagine that I collect this data today from the herd software. So this would be a cow that today is about 110 days in milk, and she was bred for the first time at 20 days in milk. So you can see that this herd has no control relative to the management of first AI. They start breeding very early, and it takes them a long time to breed their cows. Okay, so too much here. Let me show you a herd that manages first AI really well. They have a plan. They say, my voluntary waiting period is 50 some days. They start at 50 some days, and they finish at 70 some days. So in 3 weeks, they breed 99.5% of the cows, and then there's some cows that get missed for whatever reason. So this herd does a much better job, and this is what a lot of producers do today. They control the interval from when they start and when they finish first AI. Just to show to you that not every farm in the US only breeds cows through timed AI, which seems to be sort of a notion out there because people read scientific papers. So this is a figure of what happens in the US industry relative to breeding strategies. SAI means synchronized AI. Okay, so these are cows that receive timed artificial insemination. You can see that there are some regions of the country that synchronized AI represents more than 30% of the breedings. But there are lots of other places that synchronized AI represent less than 30% of all the breedings. In fact, it's estimated today that about 70% of all AIs are done on estrus, and about 30% are time inseminated. And you can see that in general, pregnancy doesn't change whether I use timed AI versus insemination on estrus. Okay, there are some cases that timed AI can create an advantage, particularly in first insemination. But in general, we expect to have the same fertility whether I use timed AI or AI on estrus. So that's why producers want to look at rebreeding cows that spontaneously come back into estrus. And they want to have tight inter-AI intervals. So let me show you an example. So here is days postpartum at the first insemination in this herd. The mean is 63 days, but you can see that they start here at about 55 days, and they finish here about 75 days in milk. Okay, so they have a short window for first AI. If you look now at the second AI, there's a little bit more spread, but the mean is 95 days, so about 31 days later. If you look at the third AI, 126 days, so another 30 days later. So you can see that every 30 days, cows are being bred because they either return to estrus or I call her open and she is re-inseminated 10 days later. Lastly, I just want to touch on something that the industry has adopted heavily in the last 10 or 15 years, mostly in the last 10 years, okay, which is genomic selection. So here's a diagram of traditional genetic selection, progeny testing. So we would identify bulls that are sons of cows and bulls that are considered superior. Okay, so we would identify these bulls from better herds that had great cows and great sires, okay, that used great proven sires in the past, and we would select those young sires to undergo progeny testing. We had no idea the genes responsible for a particular trait, and I'm going to use the example of milk production because that was an important trait selected in the past and still is today, obviously. but we expect that bulls from superior sires and dams would produce superior daughters. So a young sire, once he got into the breeding program, if he came from superior sires and superior cows, would probably have his progeny to be better than the average of the population. So there would be an estimated genetic value based on parental average, okay, if we had good records, okay? Eventually, we would learn which of those bulls were truly superior based on the progeny of those animals. So their daughters would be born, we would measure that, and eventually we would know the true genetic value, okay? So the accuracy of the information would improve. In other words, the reliability would improve as we accumulate more daughters in more herds. This would take, typically 6-7 years to gather all this information. And then came the genomic revolution. And I call this revolution because it was adopted faster than artificial insemination was. So it took not very long until the industry adopted and producers adopted. And this brought major advantages, one of which was the reduction in generation interval, okay? So it cut the generation interval from perhaps 6 years to now less than 2 years. It improved accuracy of prediction, okay, and allowed us to identify the genes, maybe not the genes, but the SNPs or the regions in the genome that are associated with particular traits, good traits, but also with bad things, such as animals that are carriers of recessive haplotypes or recessive genes. Okay, that confer perhaps bad things such as disease, but in case of reproduction, there increased risk of pregnancy loss. That also allowed us to select on the female side more heavily, not just on sires now. So now we can select daughters, and that's what a lot of farms are doing. So let me just show you one example, and I'm sure you have your own examples in Israel, but these are haplotypes or chunks of DNA in the genome of, in this case, Holstein cattle that are associated with increased risk of pregnancy loss. So there's, to my knowledge, at least 6 of them today for the Holstein breed. Maybe there are more. This is the frequency of the population. This would be heterozygous animals. And this is the estimated reduction in pregnancy per AI if you have animals in the population, because if you cross an age-1 heterozygous cow with an age-1 heterozygous bull, there's a 25% probability of a pregnancy loss in those animals, okay? So by having genomic selection, we can identify those animals and hopefully read the population of them from those animals. There's a famous bull that is perhaps known to have introduced one of those haplotypes in the Holstein population. And this is our Linda Chief that represents more than 10% of the genome of the US Holstein today. There is one in the Jersey breed called Chocolate that introduced one of the haplotypes that causes pregnancy loss in the Jersey breed. So now we know those things and we can avoid that, hopefully, as we learn more and more. There are lots of choices. Now we can select for increased productive performance at the same time that we increase for improved daughter fertility. So here is one of the traits we use for daughter fertility called daughter pregnancy rate. And this is the summation in kilograms of milk solids, so fat plus true protein, in terms of predicting transmitting ability of those sires. And you can see that there's lots of sires that confers daughters with superior ability to produce fat plus protein, and they have improved reproduction. So for example, this bull here has about a 70 kilograms increase in fat protein combined, and the daughters of this bull are expected to have a pregnancy rate of 4 points above the mean of the population, the reference population, and 4 points in pregnancy rate imply a reduction in 16 days open. So whether we use young sires or we use proven bulls, now we have choices to select for multiple traits and not cause depression and fertility. Let me show an example from our own farm here at the University of Florida. We have about 500 cows. And when I came in, we changed how we select sires, and we put a lot of emphasis in multi-trait selection program. So this is one of the index that we have in the US Holstein population, lifetime net merit. And you can see that we've improved that at the same time that we maintain protein yield in terms of breeding value. We put a lot of emphasis on daughter fertility, and we put a lot of emphasis on productive life or longevity of cows. So just by selecting better sires, we can alter the genomic architecture of the population that hopefully will result in better phenotypes. That's what we want to capture, okay? And I think that's part of the positive story. So let me show you the same way I show you the best cow phenotypically. Now let me show you some sires that genotypically are the epicenter of the breed. So this is, I visited a bull stud a month ago, and I took this picture from the #1 sire in the US Holstein population today. It's this bull here, Pine Tree, Tennessee. And what I mean by #1 sire is based on this index here, lifetime net merit dollars. Unfortunately, this bull is not positive for daughter fertility. He has lots of wonderful traits, but reproduction is right on the average to below average. But we have lots of other choices. So here's a bull that his cow conception rate, so the daughters of this bull, once they become lactating cows, are expected to have 5 points above the breed average for pregnancy per AI. So in that particular herd, if pregnancy per AI averages about 35%, the daughters of these bulls are expected to be at about 40%. At the same time that it's positive for production traits. US producers are not going to select against production anytime soon. So we want to combine the beauty of multi-trait selection. Here's another example that has positive fertility and high production and high productive life. So there are options now to change the architecture of the breed. So just to finalize, sorry for being too long, most producers have developed a plan for reproduction. They have a plan for transition cow management. So they establish that and they work that with their staff such that it's properly implemented. They have a plan for their reproduction in terms of first breeding and subsequent breedings. So they know exactly what's going to be done at the first AI and the subsequent AI. And they have incorporated a genetic program now that focuses not only on production traits, they definitely don't look much at type traits anymore, but they put a lot of emphasis on fitness traits such as fertility and health. So there is a balanced program to capture, because obviously this is cumulative in the population. Obviously, as all things that we do on farms, that's a team effort. So it's not like one bullet solution. It takes a lot of work, and it takes a lot of people. So the consulting team, the management team. But the key is when we look at farms that do really well and farms that do not very well are probably the important aspects, such as having healthy cows, having proper diet, nutrition, and management, having a sound reproductive program, and selecting for fertility at the same time that production is still a key point in the genetic selection program. Well, I thank you for your attention. Hopefully it's not too long and too boring, but hopefully there will be some time for discussion and debate. Thank you very much.
Thank you very much, Santos. Now it's time for a question. Someone want to start?
Yes, Yaniv, do you hear me?
Yes.
Yes, yes.
Jos'e, very nice talk. I enjoyed very much. I have a couple of questions to you. Can you explain how a disease that occurred within the 3 weeks postpartum is affecting fertility, insemination, and embryo development 2 or 3 months later? What is the mechanism? How it happens?
Yeah, so probably multiple mechanisms, Dave, and I'm not going to pretend that I know them in detail because it's quite complex, but I can tell you some of the things that we are working on and colleagues here, my students, that we think of. So disease often leads to hyperthermia, okay? So many of the cows that have acute mastitis, acute metritis, they undergo a few days of hyperthermia. And obviously through the work from Israel and a lot of the work done here in Florida and other places show that hyperthermia damages the early follicles. So if we think about a cow that will be bred 70 days in milk, 80 days in milk, okay, those early follicles, primary follicles with just a layer of granulosa cells, they are developing weeks or months before that first insemination, okay? Disease causes destruction of the reproductive tissue, okay? So if you think of metritis, there's massive destruction of the tissue. And I know that in early lactation, the cow wipes out all the superficial epithelium, but there is damage to the stromal cells and to the glands, to the endometrial glands. And we have induced inflammation in the uteri of heifers, and 6 months later, the transcriptome of the uteri is still quite altered relative to an animal that didn't develop an inflammatory response. So there's massive change there, probably because there is a massive change in the cell population that is present in the tissue with migration of immune cells, lymphocytes, macrophages that populate the glands. Obviously, the products, the famous pumps from bacteria can affect the central nervous system, the work that you and Yaniv have done and others looking at bacterial stressors, LPS, affecting release of gonadotropins that can alter resumption of astrocyclicity, that can affect when cows ovulate. And there's really good data showing that if a cow doesn't ovulate early on, or if we synchronize a cow and she doesn't have a corpus luteum when she is undergoing development of the pre-ovulatory follicle, in other words, if that follicle develops under low to moderate progesterone concentrations, that affects the oocyte. Okay, and so if we have more anovular cows because we delayed ovulation, now when she comes into estrus for first AI at 60 or 70 days, maybe that pre-ovulatory follicle developed under low concentration of progesterone, which exposes that follicle to excess of LH pulse frequency and affects the oocyte. And then there's all the pro-inflammatory molecules that come from mobilizing excessive tissue. So if you think of probably all species that I am aware of, as soon as we have disease, appetite drops. Yeah, it's a conserved mechanism across any species that you look at. My mom, if we were sick, she would force feed us. She would give us something to eat. But nature is telling us that maybe we shouldn't eat when we are sick. Yeah, and the cow is no different. She doesn't feel hungry at the same time that she's using nutrients for other functions. So that creates additional catabolism. And there's very good data, and I think some from Maya and some from Michigan State showing that extensive catabolism changes the architecture of the adipose tissue, induces infiltration and alters the phenotype of macrophages that produces inflammatory molecules. So now this cow undergoes a period of perhaps moderate inflammation, more so than a healthy cow does. And all of this combined with probably many other mechanisms that we have no idea somehow affect reproduction. But there's the other story that a lot of the repro data is based on association, not necessarily experimentation. Yeah, there's some cool mechanistic studies that we induce inflammation, we measure things, so we can attribute cause and effect. But the reality is that the bulk of the population studies are from epidemiological studies, like many that I showed you. So we don't know if the cow that is prone to have disease is also a cow that's prone to have less than ideal reproduction. So maybe those things work combined, and it's hard to tease that out because producers won't let me induce metritis in 500 cows to study that, unfortunately. I wish I could, but we usually do small experiments because they're quite expensive. But yeah, it's a complex scenario. It probably affects the ovary, probably affects the oviduct, probably affects the endometrium. And we have some idea, but we don't know very well all the potential mechanisms.
Thank you.
So the goal is still to prevent the disease.
Yes, that's for sure all the time. Wait, someone else want to ask? Okay. I have a question. I want to ask a little bit from the sire side on this time about those haplotypes for the fertility that you showed. What those 5 or 6 haplotypes, HH1 and so on, what do you think the impact of those haplotypes on reproduction? And again, if you are looking also on the sire fertility and how many strengths or importance you give it also to the sire fertility when looking at reproduction?
So, you know, I have a once a month meeting with a group of geneticists in the USDA because I'm involved in the feed efficiency grant with them. So I asked those things to people who are much smarter than I am, and they found that through serendipity. Yeah, they couldn't find any animal that was homozygous for those HHs or HJs in the or JHs in the Jersey breed. Yeah, so because going through thousands, today millions of genotype sequences that are available at the USDA database, they couldn't find animals that were homozygous. So by default, they assume, hey, this is a recessive lethal gene, okay? Because nobody is alive genotyped carrying that particular combination of alleles. So they define that. And then a group, Illinois and UC Davis, tracked to one of the sires for the HH1, which is Arlinda Chief. But if you look at the benefits that some of those sires brought to the Holstein breed, like Arlinda Chief, improved production substantially, it create lines of animals. I think most people would say that the bad things that came in terms of reproduction were outweighed by the good things that came relative to production. Now we are much smarter. We can identify at least those that we know and select against that. And that's what producers do. Like, for example, at the University of Florida, we have genotyped about 2,500 animals in the last few years. Every heifer born is genotyped. So when we select sires, we do have plenty of HH5 and HH2 heifers. I think today we might have probably 40 or 50 animals that are carriers. So what we do is we don't select any sire that carries those alleles. Okay, so we basically segregate against those animals when we buy semen. We tell ABS or whatever the semen company select sires, we don't want sires that are carriers for these recessive genes. And I think probably a lot of producers who genotype cattle are doing that. They are excluding those sires as choices for their breeding program because they are still being incorporated into the breed. The bull studs are not necessarily killing. If Tennessee is a carrier of one of those haplotypes, but because it has such a genetic value, that animal is going to be in the bull stud. They're not going to kill that calf. Okay, so as a producer, I have the ability to not use that animal. And I think that's what a lot of producers who genotype are doing. They are selecting against that. Now, what impact it has, it depends on how frequent that is in your population. As if I have only 1% of my animals that are carriers, the impact would be minimum. But if I have 50% of the animals in my herd that are carriers and I choose to use an heterozygous bull, then I have a high probability of having more pregnancy losses. It's just a matter of combining the right alleles that will happen, supposedly. Now, relative to the male side, I have to be honest, Yaniv, I know nothing about male fertility. The only thing I know is that when we buy semen, when producers buy semen, they look at whether that sire has high conception. The sire itself has high fertility, so they can get more daughters from that bull. So yes, that is something. But to be honest, I have a colleague who works on Jersey and Holstein male fertility from the genotype point of view. He left Florida, he moved to Wisconsin now, Francisco Peña Garicano. And the bull studies ignore that. There is no selection for the male side in terms of sire fertility. It's all based on phenotype. Okay, there is nothing. The Holstein breed or the Jersey breed today doesn't implement any genetic or genomic selection for semen quality or sire conception. It's all based on whatever the phenotype is. We use more of the bulls that generate more pregnancies. We use less of the bulls that generate fewer pregnancies. That's basically the state of the industry today.
Okay, thank you.
I have another question, Yaniv.
Yes.
Jos'e, what is the percentage of farms in USA that use pedometry? In Israel, most of the dairy farms use pedometry, and I think that as a result of that, OVSync or timed AI is not so popular here. So what is the statistics in USA regarding the use of pedometry?
Yeah, so I don't have that figure, Dave. It's increasing. Both pedometers and accelerometer collars, accelerometers like SCR is quite popular. It's increasing for two reasons. One is that as farms consolidate, producers want to know where the cow is because of mixing in pens. So as soon as somebody expands the herd, let's say somebody has 1,000 cows and they move to 3,000 cows, the first thing they do, it's kind of interesting behavior. They stop doing DHI testing. They stop doing the monthly milk measurements. They install a daily milk production system and a cow identification system. And they stop doing the monthly testing so they don't have to spend the money with that, which is bad because it doesn't contribute to the national herd information. But they do that because they spent the money on the other technology to measure milk and to identify the cow. And once they install either pedometers or accelerometers, they start using that technology for estrus detection. It depends on the region. A lot of the industry in the US is regional. If you go to the Midwest, a lot of producers are implementing pedometry and collars for detection of estrus and identification of cows. If you go to the Western states, California, Arizona, Nevada, then estrus detection is still heavily done by tail painting. Cow comes out of the parlor, gets locked up in the pens. She comes back, somebody paints the tail and reads the chalk and determines whether she is bred or not. She should be bred or not that day. So there's a little bit of regional differences, probably because in the West there is more outside activity for the cow. The freestall barns have a lot that the cow can walk and jump and express better estrus behavior, whereas in New York, Wisconsin, cows tend to stay more indoors, and therefore they rely more on the technology to detect estrus. But there's more and more adoption of those technologies.
I have a question, Jos'e. Do you think that there's enough research attempts these days to find new solutions for nutritional manipulations to increase intake at early postpartum?
Yeah, that's an interesting question. So that's a little bit of my frustration. A lot of the research today is, at least in nutrition, is industry funded. Yeah, we live a time that money is scarce and research is just too expensive. So I guess the short answer is no. We don't have enough research to understand mechanisms that control intake. Therefore, we don't know exactly what to do in early lactation to make cows eat a little more, other than, common sense and, don't overfeed low quality fiber, don't overfeed starch, don't overfeed unsaturated fatty acids, those types of things that we know are hypophagic. But really good research that answers fundamental questions, I just don't see that being done. And I think it reflects the funding mechanisms, the imagination of the scientific body that's less and less. Our students, I see that they have less and less imagination in terms of designing experiments, thinking of hypotheses. They like more to execute tasks and do demonstration type of experiments as opposed to hypothesis-driven experiments or challenging. And our funding bodies, at least in the US, won't take chances. You almost have to have the results before you apply to the grant funding mechanism. So they fund you. If there is a small chance of no success, they tell you, oh, what if this doesn't happen? They don't fund you. They don't take chances anymore. Like when I was on my sabbatical in Australia, I spent a lot of time with faculty members who had just retired, and they came mostly from England, and they were rumen biochemists. They were doing really discovery research, discovering VFAs, things that we take for granted today, but 60 years ago, we just didn't know very well. And they would take lots of chances. It was a big gamble. Pretty small experiments, but a lot of thinking and challenging. They were not afraid of, if an animal dies, it dies. Today, I finished an experiment here about 8 months ago, and the experiment cost, between the experiment itself, the student, the fees for the cows and everything, $250,000. It's like the price of a house. It's ridiculous. So yeah, it's a frustrating area. So it will be a slow progress because there's not a lot of good science being done in that part.
I would like to take a question. Okay.
Jos'e, I want to ask you about the uterine diseases versus the non-uterine diseases. Do you think that they have common mechanisms that depress fertility, or each disease has its own way or its own process how to reduce fertility? I'm asking that because in the past we found that timed AI is improving fertility of mastitic animals, but cows that have had metritis postpartum were not affected by timed AI. So what is your idea about that?
Yeah, so I think there's both sort of a conserve mechanism in place, common mechanism between the two, probably through the systemic inflammatory response that both can elicit. Maybe pro-inflammatory molecules live in the uterus, pro-inflammatory molecules live in the mammary gland. Let's say if you have more of an acute mastitis case with LPS or peptidoglycans getting into
cytokines.
Yeah, cytokine storm, Yeah, that's exactly right. So, but I guess in my small uterine world, I like to think that uterine disease are more devastating because it not only causes those responses, it causes a major cytokine response, a major acute phase response, release of fibrinogen, release of acute phase proteins, haptoglobin, seroamyloid A by the liver, but it also damages the tissue. So, and that damage doesn't get recovered very easily. We think that, hey, the epithelium gets reestablished and life goes back to normal. There's some pictures in old papers in which people looked at cows that had disease or not, in this case, metritis, and there's this massive accumulation of lymphocytes in the glands. It almost occludes the glands. I like to think that that is not very good for a pregnancy. It probably secretes lots of molecules that are pro-inflammatory, maybe alters the tolerance mechanisms to pregnancy, like the tolerance to alloantigens from the male side, and it probably affects the ability of the glands to secrete estrotroph. So I think there's both, Dave, sort of common mechanisms through the systemic inflammatory response, cytokine response, pro-inflammatory molecules circulating in blood in the more acute cases. But there is also the localized effects of, in particular, relative to reproduction, of an inflammatory response in the uterus that damages the tissue, and that damage may not be recovered fully through therapy and time. Yeah, they say that time fixes everything, even a broken heart, but maybe not a damaged uterus.
Okay, Vika, you want to ask?
I would like to ask another question regarding the efforts. We didn't talk about them today. And we recently experienced in Israel a reduced fertility in efforts. They don't have mastitis, don't have metritis. They should not suffer from
like
stress.
Virgin heifers, growing heifers.
Sorry?
You mean like 14, 15-month-old heifers?
Yeah, replacing heifers, yeah. So the reduction is coming in a waves, about every 5-6 years, in the winter and in the summer. And I would like to ask you whether you experience such a phenomena in the state or another place, or in general to talk about a little bit the heifers fertility, because we consider it the reproductive potential of the herd.
Yeah, well, Zavia, I'll be honest, like we haven't had that characterized. We have sporadic problems on a particular farm, but as a herd or a discussion, my world is small. I go to 4, 5, 6 farms. I don't go to 35,000 farms in the US. So I don't know what the picture is, but as a population from the database, fertility in heifers has not really changed. In the US today, just to give an idea, pregnancy per AI with conventional semen for all inseminations in heifers averages about 56%. The first AI is usually better, but as a nation, almost 3 million heifers a year that get bred for first time, that averages about 56% for all AIs, okay? So we haven't seen a drop over the years. And from my small experience of going to a few farms, usually that's a farm-specific problem. It happens. A lot of times we don't figure out what the problem was, whether it was an animal problem or a human element problem, something like that, and then the problem goes away. But it's never a big concern. Now, there's been a change in the industry in the last 2 or 3 years, and I'll tell you a little bit what it is that may affect our numbers in the future. Heifer values have plummeted in the US in the last few years. I told you that if I raise a heifer and I sell her as a pregnant animal at 23 months, I lose about $300 per animal. it costs me $300 more to raise that heifer, get her ready to calve, than what I get if I sell her in the market. So it's a bad business to raise heifers to sell to a second party, because it's a 2-year investment to lose $300. So what producers have done, and that's been a major shift, is to change the breeding policies. So now we have herds that 100% of the heifers and 100% of the first lactation animals receive only sex semen. So something that we would never do 5 years ago, today is normal. You walk in a farm, all 13-month-old heifers, they are bred only with sex, with X-sorted semen. And all first lactation animals are bred with X-sorted semen. And the rest of the herd, they are bred with Angus semen or Wagyu or some some beef breed. And the reason being is that a newborn Holstein heifer, if I sell her, if she's born now and somebody comes to pick her up, she's worth $20. And a black crossbred calf, born male or female, if it is born, I sell for $180. So it's an economics number. So it is possible that if we have this conversation a year or two from now, that we're going to see a slight drop because of more sex semen being used, and you get slightly drop in fertility, maybe 85% of the conventional semen, something like that. But that's a major change in the industry now. Everybody has, you go, just to give an idea, I went to ABS, and ABS in Wisconsin has a lab with 136 flow cytometers working non-stop, 23 hours a day, 6 days a week, sorting semen to supply the US market. So it's just a ridiculous thing. When I saw 136 flow cytometers in a huge room to produce, I think maybe 5 or 6 million doses of X-sorted semen for the Holstein breed, that has really changed how the industry goes and moves.
Jos'e.
Thank you.
Yes, Hen, how are you?
Thanks for a great talk. I would like to shift you back to the nutrition side, if I may. How much do you think negative DCAD programs have contributed to this reproduction success?
It's very hard to pinpoint one thing. Yeah, like maybe if I'm doing research with that, like I am, I like to give some credit to it. I think for the multiparous cows, the cows are going to start their second or greater lactation. Based on what we know today, formulate those diets that are acidogenic prepartin brings a lot of benefit in terms of health in early lactation, in particular metritis, uterine disease in general. So there's probably some role relative to the subsequent reproduction. Can I give you a number? I have no idea because we just don't have those data. I can tell you from the research that's available and from on-farm observations, I like to say that producers almost always observe right, but many times they interpret wrong. But it's very common for a producer, you walk in, they say, hey, my transition cow program is not doing very well. Let's say if it is DCAD or acidogenic diets, the urine pH is too low or too high. So let's say urine pH is in the last 2 weeks average below 6, 5.5 or 5.4, or is 7.5. They always observe that they have more postpartum problems. So it's a very common thing that happens. I don't know if it is just automatic or it's a true observation, but it seems to be a real observation. So
how popular are those programs in the States?
In big farms, almost every single one uses. Depends on the size of the farm then. So all the farms that I visit, they use it. In small farms, when they cannot have a true transition program, or they don't want to manage the diet in terms of monitoring urine pHs, doing chemical analysis routinely in forages and byproducts, those types of things, then oftentimes they use other routes. They give calcium after calving, they give subcutaneous calcium. Some are using aluminum silicates now, not very popular, but there's some use being incorporated. Based on the national statistics, about 40% of the farms use acidogenic diets.
Okay, there's more question for someone?
Yes, Yaniv,
Yes.
it's Israel.
Okay.
Hello, Jos'e. First
How are
of
you,
all,
Israel?
thank you for your excellent lectures as usual. And you will not be surprised if I will take you to the heat stress topic. I just submitted an article, first of all in Hebrew for the ICBA magazine about fertility in heifers. We talked about heifers. It's not exactly as Roth said. It's the decline is winter and summer, but conception rate in winter dropped in last months in 5%, while in summer it dropped between 10 and 15%, and this is a big difference. Now, I faced this same problem almost 10 years ago in Vietnam, where heifers suffered such an extreme heat stress that we put data loggers and they were hyperthermic, and they invited me exactly because heifers didn't get pregnant. And now the question is, if climate is warming here in Israel and heifers started suffering from heat stress, or there is another reason. Anyhow, I conclude my article saying that we will have to analyze if it is economically justified to cool heifers in order to get 10% more conception rate in summer, when we know that most of the heifers, we try to concentrate them to those months that will give us more calvings to the next summer. So half of the problem is already solved by not having the necessity to inseminate cows in summer, heifers. But those heifers that are inseminated drop in 10 to 15% in last 3 years. I discussed it with Yoel from the AI company, and he confirmed it.
Yeah, so that's. I'm sure that I have no doubts that heifers suffer from heat stress, and we see that here in the summer. Conception drops in the heifers. It doesn't drop as much as it does in cows in terms of absolute values, but it does decrease in heifers. Like I mentioned to Zivi, we haven't seen this pattern of decline in fertility in heifers, or at least we have not characterized. So I have to say that maybe it hasn't been characterized, but we haven't seen that as a complaint or as a topic of discussion. You make a great point of this thing of heat stress. We have this sort of dilemma, yeah? In a lot of places, heifers are raised outside, either in dry lots or they are raised in pastures or in paddocks. Depends on the region of the country and the time of the year. And the average person out there, we have societal pressures. They like to think that an animal that's outside is better for the animal. Yeah, that's very common. I'll give you the example of Florida. You show a cow on pasture today, everybody will say that she's a happier cow than if I show a cow on a freestyle barn lying on a sandy stall, a clean bed with sand. That's the perception of the population. At the same time, it is miserable. It is terrible to be a cow in Florida in the month of July in a pasture, or a heifer, But we suffer from these societal pressures that they want us to do this, at the same time that we cannot control the environment if we go back to, pasture systems or this types of situations. So I don't know where our industry will go if we're going to get to the point of discussing better infrastructure and facilities for heifers, which is needed in a lot of places to combat heat stress and other things. I can give you examples of some places that suffer from blood-borne parasites such as Babesia and Anaplasma. So once they go outside, they get ticks, they get biting flies, and then it breaks with anemia and fever and problems like that. So having facilities where you keep animals indoors and cool them would be ideal. But we have this debate of where is society going to tell us or fight with us relative to normal procedures on farms, and that is a battle today. So I think if you were to ask where is the average producer in the U.S. going in relative to that, he or she will probably wait to see what's going to happen before they invest money. Okay, that's basically what they're going to do, because they just cannot read between. They understand I'm pretty sure they all understand that those heifers suffer, and that's a problem, and it probably affects reproduction, and it probably will get worse with global warming. But the uncertainty probably will create some caution in where they invest their money. And heifers in general are not necessarily a big priority, to be honest. Once they leave that hutch at 60 days of age and mortality and morbidity is low, they drop to the bottom of priorities.
I even don't, I'm not sure that it is even cost effective under Israeli conditions, which are much better than US conditions, economical conditions. It is something to be studied, but we started facing this problem in last years, and then I think that we need to study it more profoundly.
Like if there is an area that is completely neglected in literature, it is a heifer once she is weaned. Nobody does research between 70 days of age all the way to first calving. You count your fingers in the world.
But in this article, I refer to an article, a recent article, where very short cooling around time of insemination gave significant improvement in fertility. So in good management, you can let's say solve great part of the problem.
Okay.
Getting late in Israel.
Question? Yes, getting late. Yes, I think then we want to thank you on the great presentation and for being with us tonight here in Israel in your day. And I want to thank everybody that came. And again, we want to continue to make those webinars. Then if someone wants to suggest a person to invite or to raise the topics, that you can talk to me and we'll try to bring new webinars. And I want to thank everybody. Thank you, Jos'e.
Well, have a good night.
Thank you.
It's a pleasure. Good seeing everybody.
Thank you very much. Thank you.
Thank you.